Welcome to the second in the sequence of 4 of my lectures on quantitative methods. In this lecture, we are going to start using R in a somewhat serious way. That's not the point of today, though, but we're going to use it as a tool to think quantitatively and visualize data and formulate quite concrete versions of a conceptual model. The point of today is something I made reference to before, which is that if you are clear and specific enough about the model that you have in mind when you come into your data analysis, you actually will have the same tool you need to analyze your data as you need for simulating data before you've even paid for and carried out your research project. So let's be more specific about what I'm getting at. I want to turn off annotations. So you remember what a data generating process is, and I've emphasized how important it is to have a model and think carefully about your model and be transparent about your model that you have in mind in order not just to carry out your analysis, but in fact that you need to do that at the stage when you're designing your research. And I've also mentioned how important it is when you're designing your research and planning your analysis to think about what the sources of variation are and indeed how much variation there is. And that's going to become really critical today. We're going to simulate data as a way to get insight into whether our research design is going to be sufficiently powerful in a statistical sense in order to answer our research question. Right? So if you have a research question that you're given and you need to know whether it's going to cost $1,000 or $1 million to answer that question, you're going to have to go through essentially all the process that I show you today. In other words, you're going to need to think, what is the natural variation in the stuff that we want to measure? How noisy is it? What are the sources of natural variation as compared with the variation that we are interested in? And then with the help of a model, can our accordingly, let's say with a given sample size, which is often the question that you're answering when you are figuring out whether you are going to need $1,000 or $1 million, is how much sample, what sample size do you need in order to have enough statistical power to answer your question? Okay, so let me run with this example for today. We're interested in the effect of pesticides on bees. Now, I know nothing about this really in terms of a specialization, but of course we all know that certain pesticides have had enormous effects on wild bee populations. So, but let's pretend that our plan for figuring out how impactful pesticides are on bee populations, our plan is to look at the change in population of a beehive over 1 year, taking into account how far away it is from, say, a farm which is using pesticides. So in fact, what we're going to do, rather than going and looking for beehives, is we're going to place them. So this is sort of experimental. We're going to find homeowners who are willing to host a beehive for a year. And then we're going to look at how far each of those sites is from a pesticide using farm. And we're going to look at how the populations of each hive change over 1 year. And the question is going to be, to what extent hives that are closer to the pesticides suffer a larger population loss. So we have a conceptual model that's very simple. the causal model is just two different things are going to affect the population size in each hive. And one of them is the exposure to pesticides, so the distance from pesticide source. And the other is going to be all the other hive-dependent factors which may make a hive grow in size or shrink in size. And so that we don't have to know what the details of these are, but the key thing here is that they're hive-dependent. In other words, they're not going to be affected by how close the hive is to a farm. So you can think here, for instance, is maybe the fitness of the queen. So we're going to come along and seed each of these, start each household out with a hive, and for some natural reasons, some hives are going to thrive more than others. So in fact, let's go a step further and formalize this conceptual model. A better word than formalize is to parameterize. So we're going to take it from these arbitrary forms of these relationships. These are causal, but it doesn't say sort of what shape the relationships have. Let's actually formalize it with an equation. So we can say, let's assume that the population after 1 year in a given hive is equal to the initial population. And maybe we control this. So let's actually make it the same for every single farm. Okay, so we are going to come and actually fund and transport these hives to each household, and each one is going to start out with exactly 1,000 bees. And then a term here, which is related to the proximity to pesticides. So 1 divided by the distance, so the inverse distance, in other words, how close the house is to a pesticide using a farm. And then a factor, which we can call alpha, which is the strength of the effect of closeness to pesticides, or the effect of pesticides on population. In other words, this is the thing that we actually want to know. The whole research project is really about finding out what is the value of alpha. How much effect does proximity to pesticides have on population changes? Okay, so if pesticides are bad, by the way, for bee populations, then alpha will be a negative number, and if pesticides are very bad, then this will be a larger negative number. Okay, and there's another term here, and this is really a critical part of this kind of simulation. This is the factor for all of these other hive-dependent impacts on population change. And so, as we're going to see, this is not a single number that is the same for all hives. This is a random variable. This represents a distribution, which is the natural variation or variance in hive growth. In other words, what would happen to a hive if alpha was 0, or if there was no pesticides in play at all? So if I just set 1,000 hives out, if I just set a number of hives out, each with 1,000 bees on day 0, and had no pesticides involved, then at the end of a year, the hives are all going to have different sizes. Some of them will be a little bit more than 1,000, some might be a bit less, or maybe they all be a bit more. This is going to capture that variation, that natural variation in hive population change. Okay, so in fact, this is hopefully what all of these definitions here tell you. Okay, so again, how big are beehives? Without being expert, I've sort of made some assumptions here that our hive physically is sized appropriately for 1,000 bees. So in fact, I'm going to show you in a moment that I'll assume that on average, if I laid 1,000 hives out and there were no pesticides, that after a year, the average size would still be 1,000, but some of them would be 900, some of them would be 1,100, and so on. And there's going to be some natural distribution, natural variation. So how do we find out what that natural variation looks like? This is something we have to do before we design our research. as you will see, we need to understand this natural variation in order to know whether we're going to be able, whether our research is going to be successful. In other words, whether we have a large enough sample size, or maybe whether we need to put hives closer to farms than we might easily be able to do, or whatever. So in order to actually be able to have confidence about whether our design is going to be able, statistically speaking, to answer the research question, we are going to have to investigate what the natural sources of variation are in advance. Again, you can think about a lot of this design stuff. What it often comes down to is choosing a sample size. So, as I said, let's think about what would happen if we set some hives out for a year without any pesticides. And now let's get the help of our scientific calculator, which is what you can think of R as, in order to start actually simulating or quantifying and thinking about these things quantitatively, and then simulating them. So let's this is now what we call syntax or code that we can either type into R, or we can type into a script, a program, a text file, and then tell R to run it. At this point, by the way, let me mention that if you go to the RSets tools page, you will find links to the exact presentation that I'm showing you today under this synthetic data lecture. Here's the material that I'm sharing, and also the code that I'm running bit by bit throughout the lecture. You can download all that code right here and run this in R step by step. So here's the first little bit of code. I'm going to set P0, this initial value, the size of the hive, to 1,000. And then NBs is going to be the number of bees, the population of the hive at the end of a year. Oops. And so as in our equation that we were just looking at, it's going to be P0 plus, and then this is the epsilon term. For a moment, there's no pesticide term because we're thinking about the natural variation. Now, what is this? So this is a function that's built into R that gives us a distribution. So this is the normal distribution, and it actually says take 10,000 different draws, different samples from a normal distribution that has a mean of 0 and a standard deviation or a width of 100. Okay, so what I end up with here is that NBs is in fact not a single value. It's 10,000 values that come out of this equation. All right, so this R norm function gives me 10,000 samples from a normal distribution. Now this next line, well, let's ignore that for a moment. I'm then going to just plot a histogram of all of these 10,000 values for NBs. And the rest of the stuff is just choosing how the histogram looks. XLim here is just telling me how much to show on the x-axis. I'm going to just show an x-axis that's from between 600 and 1400, and I've just defined that there. By the way, this is now a detail of language you can worry about later, but if you want to tell R that some variable, in this case XL, has give it a list of numbers as the value, you use this C for concatenate operator. So this just means store this list of two numbers, 600 and 1400, in a storage box called XL, and then here I'm using that XL to specify a parameter that modifies how this histogram plotting function works. And this tells me how many bins I want in a histogram. Remember, I told you now I can not, these are both optional. I could just ask to give a histogram for NBs with default values, and you can try that. Remember that I told you your statistical software is always going to choose the number of bins in histogram by itself if you let it, but you should play around with them a little bit because you can get different qualitative results in a histogram with more or less bins. So in this case, I'm just specifying for consistency that to have 50 bins and the range I want to look at. Okay, so that was all an aside on our statistical language R here. But what have we got? We have plotted 10,000 draws from a normal distribution. So not surprisingly, the histogram looks like a normal distribution. It looks like a bell curve. The mean value of the bell curve is 1,000 because that's that's our starting value, and this distribution here had a mean of 0 spread around it. And the width of this thing has a standard deviation of 100 Bs. And as you may remember from your from knowing about the bell curve, 95% of the bell curve is within 2 standard deviations of the mean. So here's 1,000, standard deviation is 100. That means between 1,200 and 800 are 95% of the hives. Okay, so that's just features of this normal distribution. Now, this is all very nice, but if I'm actually planning a research project, I'm probably not going to be able to find 10,000 households to host hives. What's a more realistic number of households that I could recruit given a reasonable budget? Well, maybe I can get 10 households. Okay, so I'm going to buy 10 hives. Each hive, I don't know, how much does a hive cost? $1,500, $200, I don't know. So we're talking about some thousands of dollars here for this research project. And I've kind of, more succinctly put that whole line, several lines before, into one line. And so this is the histogram I get, where I just sample 10 times randomly from that same distribution. So this is now answers the question, or simulates the experiment where I put out 10 hives with 1,000 bees each for 1 year. And then I look at how many bees are in each hive at the end of the year, and this is the distribution of values I get. Now, you can see it doesn't necessarily look like a bell curve anymore, and that's because it's just 10 random draws. And so it's the luck of the draw. In fact, if I did that same experiment again the next year, I would get something different. And if I did it again the next year, I'd get something different. You can see that if I did it again the next year, I'd get something different, and so on. And so this kind of variation, you can see, this variation is just due to this natural features of hive evolution. This is going to give some limit to how precise a study I can do to find out what effect something like pesticides is going to have. Because if the effect of pesticides is smaller than this variation I get from repeating the experiment multiple times, then I'm clearly not going to be able to resolve the effect of the pesticides. Okay, so I want to make sure that's really clear. I'm thinking about what the natural I've parameterized what I think is going on in this in the world. This alpha is what the whole project is aimed at determining. And I'm thinking now about what the natural variation is in the system in terms of the evolution of a hive over time. This is something I'm going to control in my research project. And D is something that either I control or could be by luck, by random as well. We'll look at that next. Okay, so now that was thinking about what happens if there's no pesticide. Now let's think about what happens if we actually design this research project, given that there is pesticide. So proximity to farms, this term here matters. So what's going to be the distribution of distances D? And as I just mentioned, we could actually choose this, control this in our research design. So this is only one possibility, but one option is that we actually just take households that are in exactly 2 groups. One group will all be 1 kilometer from a farm, and the others will all be 10 kilometers from a farm. And then all we have to do is compare those 2 groups and see whether the ones who are closer to the farm have smaller populations at the end of the year, and by how much. Okay, so hopefully if we do this right, we will be able to just look at the populations after a year in group A and group B, and then back out from that how big an effect alpha is, what the value of alpha is. But we've just seen that the natural variation that comes from that's captured by this epsilon term, in other words, the natural variation in hives is going to fuzz out any effect just partly from this difference in proximity to pesticides. So the question is really, how big an effect of alpha will we be able to detect if we have a total sample size of 10? If alpha is really big, it'll be easier to detect. If alpha is a small effect, if pesticides are only a little bit harmful for bees, then it's going to be harder to detect because of this natural variation. So let's simulate this a little bit. In fact, for the rest of the lecture, I'm just going to give one example, which is to assume that alpha is has a magnitude of minus 50 kilometers. The units may look funny, but that's what it comes out to here. D is measured in the distance between a farm, and remember that this term 1 over D is like the closeness. It's the inverse distance. So so if I have two groups and one of them is 1 kilometer from a farm, then this term will be 1 over 1 kilometer. And for the 10-kilometer group, it'll be 1/10, actually 1 over 10 kilometers. And so the units of alpha here come with a factor of kilometers. But in any case, the minus here means that insecticide is going to be bad for bees. It decreases the population. And then the size of this effect here is how bad the chemicals are for bees. Okay, so I need to put these numbers together and simulate them to see whether I'm actually going to be able to tell the difference between these two groups. So let's do it like this. Plugging in, we've got group A here, which at the end of the year is going to have size 1,000 minus 50 times 1 over 1 kilometer plus the natural variation. This is the random. And group B will be the same, but 1 over 10 for 10 kilometers. It's further away. So let's simulate now what happens if we just had a total of 10 hives, 5 in each group. So we'll set n equals to 5 hives. The initial population is 1,000 each group. We're now setting, we're assuming a value for this parameter, which we're actually trying to find out. Okay, so in our project is actually supposed to figure out what this value is. And so when we simulate, we're going to say, well, what if it was this big? And then we could try a smaller number and a larger number, as you'll see, in order to figure out how sensitive our method is going to be. For instance, pretend that imagine, at the end of the project, we're going to report back to a policymaker or scientist, and in fact, maybe the question from policymakers is, either tell me that what I want to know is the size of this effect, unless it's so small that it's negligible. So if alpha is is smaller in absolute magnitude than -5, then we don't care about it. So if you can assure us that it's that small, you don't need to tell us how big it is. But if it's larger than that, we want to be able to detect, know the size, so that we can properly price countermeasures. Okay, so I'm actually specifying the value of that parameter for the simulation. And then this here is now just, again, for display purposes, this is setting exactly the bins in the histogram so that they're all consistent. And now let's calculate, as in these formulae here, what are the population sizes in each group. I'm going to have 5 hives in each group. Here's the group A. It's going to have this 1 over 1 closeness, and this one has 1 over 10 closeness. And each case, I have what we've seen before, which is this is the natural variation. It's pulled from a normal distribution. Okay, what do we get? Oh, and then I'm going to plot them. And I'm going to plot the group A, which is closer to the pesticides, in red, and I'm going to plot group B in blue on the same histogram. That's all that's going on here in these two commands. So let's look at the first one. So I run my research project and I plot my data. And each of these bars now is actually a single hive, right? So you can see one here. And so these are these population sizes of each hive at the end of the year. This is Actually, there's one red and one blue in this bin. And you can see that they're roughly centered around 1,000. So on average, they didn't change too much, but some grew larger, some grew smaller. And the question is, on average, did the red hives grow less or did they shrink more than the blue ones? In fact, I can look at the mean here. It says the mean is 906 for the red ones and 1,071 for the blue ones. So indeed, it looks like the ones that were closer to the pesticides suffered. But how confident can I be, right? The statistician says, well, okay, so what is the actual statistical difference and how large is alpha? So I know that I've actually assumed alpha is minus 50, but if I didn't know that and I was seeing these data, right? So I'm simulating the data for my project. If these are the data I found, what would I be able to figure out? Would I be able to figure out the value of alpha? What constraints could I put on it? What would be the confidence interval of my estimate of what alpha is? Well, look what happens if I run the same study the next year. Okay, what does that mean? I just press go again on all of this another time. I get this different answer, different set of populations at the end of the year. And if I do it again, I get this. If I do it again, I get this. Okay, and so you can notice that okay, so in these cases, the blue hives always have the larger mean. Here's a case that is contrary. So I ran this experiment and I found a larger average population for the red hives that were closer to the pesticides than I did for the blue. Just bad luck, if you like, or good luck. Maybe there's nothing but luck going on here, right? So I need a statistical method that's going to correctly assess how confident I am in my inference about alpha. And so I can see by doing this kind of simulation, what are, given the variation I expect in the measurements I'm going to make, how powerful is my research method in order to answer my question? Okay, so that's what I can see by looking at all those. Okay, now just for reference, if instead I had tens of millions of dollars, and I'm going to go and put out 20,000 hives. Okay, now those distributions look like this. So I've just done the same thing here, except I put n equals 10,000 here. Everything else is the same. And so now I've got 10,000 hives in group A and 10,000 in group B. And sure enough, you make an inference from this. I want you to think carefully here, though. So I've actually written the means. So we have a mean of 950 for the red ones that are closer to the pesticides, and 995 for the blue. Take a moment and tell me qualitatively or as quantitatively as you can, just looking at this plot. And you don't need to know any fancy statistical methods at this point. Just use common sense and looking at this plot and tell me what is the value of alpha? Or first of all, what is the difference? I've written numbers up here, but these numbers were not necessarily credible. They weren't. The difference was not something we had confidence in. How do I get the difference from two curves like this? And how precisely can I know them? Okay, I want you to think hard and write down a short description of an answer to those questions, right? So what is the difference between the means of these two distributions? And how precisely can I tell from these data? And if you like, take the next step, and then what value of alpha do you infer from these data? Okay, pause and come back when you've done that. So I'm just going to give the qualitative answer here. And in fact, my point is just a very qualitative and conceptual one. And that's this. Look, if I look at these two curves, I can see that they're clearly shifted, right? In fact, I've got a bell curve in each case. And I can see the blue curve is shifted to the right of the red one. They're not perfectly smooth, but I can see if I took more data, they'd probably become perfectly smooth. And so the question is, how confidently can I tell you how much blue is shifted to the right? And I would say, well, look, I mean, I can see it's just displaced here quite consistently. And I can look at my scale, and that's 50, so that's 150. I mean, yeah, it looks like roughly, maybe 50 bees. Now it's not it's okay. It's not exactly 50 in the limit. You can do the math because I've got 1/1 minus 1/10. Okay, but and I'm not going to go through that at the moment. Here's what I do want you to see. The precision with which I can tell the difference between the means of these two curves is narrower, better precision, than the width of these things, right? So the width of these curves, it looks like, is about 100 Bs. In fact, of course, it is exactly 100. The underlying distribution has a standard deviation of 100 Bs. But that's not the width of these is not my limiting resolution to tell differences between means. I'll say that one more time. I can resolve differences in means between two bell curves that is much smaller than the width of the bell curves. Okay? And you can see that when I have more observations, I can better resolve a difference in the means between these two. And sort of jumping forward to next lecture or two, in fact, the resolution with which I can discriminate the means goes like 1 over the square root of my sample size. Okay, so having a sample of 10,000 here helps me a lot, but only as in proportion to 1 over the square root in being able to resolve differences like this. Okay, again, the main qualitative thing is if I get enough distributions, I can tell quite precisely the difference between two bell curves, even if that difference is much narrower than the width of the bell curves. Okay, let me make things just a little bit more complicated. Rather than having two groups like I did here, in reality, I don't know where the pesticides are when I'm recruiting households. So here's an alternate design. I'm just going to go around and recruit a whole bunch of households, and then after I've got them set up, I can go and find out where all the pesticides and the farms are. And that means I'll have a whole range rather than just two different values of distances between farm and bees. So that means now I have another source of variation. Instead of just two groups of distances d, I'm going to have a whole range of distances d. So let me model that as well. And I'll start off again with a large number just so we can see what the distribution looks like. And I'm going to say the distances look like this. And so this is another one of these distribution functions built into R. This is an exponential distribution, and I'm using a scaling parameter 0.1 in it. There's a whole family of distributions that are built in. You can access like this without getting into the theory too much. Here's what it looks like. And I could have used something much simpler. I could have used a uniform distribution and just said, well, I expect the number of houses that are 1 kilometer away to be the same as 2 and 10 and 20, and then at a maximum of 30, and I would have just had a distribution that's a square rectangle. But this is actually a more slightly more realistic kind of distribution we expect for distance from some randomly located farms. Okay, so I have now a way to model the variation in distance of my hive sites. But once again, I'm not actually going to have enough money to have 5,000 of these. Let's simulate with a more realistic number. What if I had 10 hives? What is the distribution of distances going to look like? Well, here's a draw I get where there are just 10 hives. Okay, so I got some that are only 1 or 2 kilometers away and spread out like this. But again, if I rolled the dice again, in other words, if I just did my research project again the next year, or if somebody else did it somewhere else, they would find different set of distances. And so I could just, you've seen it a few times now, but if I redo this, I'll get a different set of distances. Okay, let's put all of this together, full model together now, where I have a distribution of distances, I have a distribution of natural hive growth, and again, I'm going to see what happens if the real value that I'm looking for was minus 50 kilometers. If the units are confusing, just call it minus 50, right? So this is the strength of the badness of pesticides on bees. And again, I'm now using a more realistic sample size that we can afford of 5 hives. It looks like I've gone down from 10 to 5. So now there's 5 hives total. I'm saving some money. Okay. And so I calculate now all of the hive populations at the end of the year. This is going to give me a list of 5 numbers. And, because I'm going to have to write a report up at the end of the term for this project. So let me put these data into a nice table so I can actually just say observations and make something called a data frame with these different values and get the house ID and the distance and then this final population all together in a nice table. And I can actually format that table a bit more nicely. There are many different ways to do that, but here's one, looks a bit obscure, but you'll find easier ones. Or maybe I want to export that nice table to Excel format, and so this just writes this table called observations to a file, myfile.xlsx, which I can load into a spreadsheet. Okay, that was all just an aside, really, of showing you some output manipulation in R. But back to the point, we're now going to simulate what happens when we have a sample of households that are sort of randomly situated, and these hives that have their own internal dynamics and suffer from being near pesticides. Okay, and actually, for the purposes of plotting again, let's think for a moment in order to understand this. What happens if I have 1,000 hives? Here is a plot of my results. Here's the distance from the hive to the nearest farm with pesticides. This is in kilometers. And here is the final population of each hive. So I've just said, I calculated P1, and I'm just now plotting D versus P1, and it creates this. Actually, maybe it would be a little bit easier to, rather than plotting D, plot that term 1 over D in our equation. So instead of distance, we'll plot closeness here, okay? Now it looks like this. So I have to find F equals 1 over D, and I'm plotting F versus that final population. So here's F. What is this? Well, if D is very large, 1 over F becomes very small. So 0 represents infinitely far away from pesticides, and 1 here represents being within 1 kilometer of pesticides, right? So I've got a scale from 0 to 1, which tells me how close I am to the pesticides. And the vertical axis is my final population. Each point is 1 observation. Okay, I've got 1,000 hives. Now, I want you to take 10 minutes and think through a series of questions. If you get stuck, you can come back for a hint. But I'd like you to pause and think about these. We've assumed a particular value for this parameter alpha, size of this effect. Qualitatively, how would the look of this plot change if alpha were stronger? So instead of being minus 50, it was, say, minus 500. Okay, in other words, if pesticides were worse for bees, how would this plot look? Second question, what if epsilon were narrower? So if that natural variation in hive growth was smaller? And third, what if we had fewer observations, if our sample size was smaller? Now, just let me start with a hint, or maybe you've already paused and come back. What are we looking for? Just to remember, in our formula, or as a general version of our formula, we're looking at the relationship between the closeness to a farm and the hive size at the end. And this is exactly the relationship that we're plotting now, is this factor versus this. And so if alpha is a strong effect, we expect a negative slope in this plot, right? So really our entire project now, our entire quantitative or statistical analysis comes down to, is there a negative slope in this plot? And my answer is, eyeballing the thing, I would say no, I really can't tell. If I ran the right statistical procedure on my data, maybe I would find out, yes, there is a negative slope here. But if you ask me to eyeball it, I'm really not sure. Okay, but the thing we're after here is going to be to estimate the slope of a best fit line through these data. Okay, with that in mind, what happens if alpha is different? What happens if, oh dear, here we go. What happens if epsilon gets narrower, and what happens if our sample size is decreased? Go ahead and answer all of those. Okay, let me just make a few more comments in case you are having difficulty with that. Somebody asked in class, I don't see epsilon on this chart. Where is epsilon? And what were some other good questions? Well, okay, so let's answer this first of all. The x-axis is precisely just the proximity to, in other words, 1 over d, the proximity of each hive to a farm. That has nothing to do with bees, essentially. It's got nothing to do with the effect that we're after. It's really just determined by where each house is. The vertical axis reflects the hive growth over the year. And the width of this, the vertical spread, is precisely from epsilon, right? So the variation comes from, at any given value of f here, any given value of closeness, the vertical variation reflects that natural variation from epsilon. And so that's what you see on the vertical scale. Okay. In case that helps, take another break and see if you can answer each of these. And now I will give a quick answer. Okay, so what happens if alpha were different? Well, alpha has a value of -50 right now. If alpha was -500, it would mean that the effect on hives that are close to pesticides is going to be worse. It'll shift values on the right of this plot downwards. And so it means that it will actually tilt this cloud of observations to be more downward sloping. What happens if epsilon gets narrower? Then at any given value, well, down here, it means that all of these observations will converge towards 1000. At any given value of closeness f, it means the values will converge towards what will form some kind of look more like a coherent line that's going maybe downwards. And if n gets smaller, it just means we have fewer of these points. It's like just randomly taking some of these away would be, there's no difference in the sort of distribution of them, but we just have fewer points with the same distribution layout. Okay. So let's get R to actually help us answer that thing which I wasn't willing to eyeball. Is there a downward trend? And so what we do here is plot f again. That just makes that plot. And now I'm going to, in one line here, do two things. One is to fit a line to the data, and the other is to add that line over plotting the figure we already had with the red color. I don't know why it's called AB line. Maybe it's like, intercept and slope or something, but that's overplot. This thing, again, these are just functions built into R. LM stands for linear model. And so this is, again, it's a bit cryptic, but this is the way to fit a linear model. And in fact, this part inside this is our syntax for specifying a model. And what does this say? It says P1, which is our population at year 1, has a functional form of a constant. So 1 is the way that it designates a constant, plus a linear term in F, which is the closeness to farms, the 1 over D. And so what this is going to do is estimate a coefficient here and a coefficient here. Well, the coefficient on the constant is the intercept for my line, and the coefficient here is the slope of the line, right? So this is C and M in your line formula. And then, it's going to plot the result. And so in this case, we have an intercept of a little bit more than 1,000. And we have a downward slope of some amount. We're not showing what the amount is, but here's a better way to visualize the same thing. Instead of just drawing that line, let's actually use a kind of fancier visualization. So again, we're making this linear fit. We're going to call that thing fit, and then we're going to give it to this other function called visreg, and it's going to show us not just the line, but also some predictive confidence interval here in shaded overlying the data. And so that we can see something about whether we're actually able to statistically resolve that downward slope or not. Okay. Now, let's go back to a more realistic sample size. Let's say we have enough money for 20 different hives. We're going and getting them randomly, whoever will let us site our hive in their house. So we have a distribution of distances. Everything's the same, except I've just decreased the sample size. So now our scatter plot looks like this. There's just 20 dots. But I can use that same procedure to fit a curve. And lo and behold, this is the final result of my research project. I get this this plot, and says, yes, well, looks like there's a maybe downward slope, but it's kind of only maybe. In fact, look, if I redid the project, I get a steeper downward slope, or this one, which slopes up. All right, and am I going to get any strongly upward sloping ones? There we go. Look, I did the one year. I redid this research project, and I got a strong upward slope. Just bad luck, or just luck of the draw. So, and you can see, in this one, is it downward sloping? Well, this confidence band is telling you actually we can't tell what the slope is here, whether it's down or flat or even up very well, right? So it's giving you a sense of what the confidence is. So is that best line the inference we're after? Well, sort of, but we can put it into numbers form. And in fact, what we really want to know is what is the slope, which is the coefficient on here, which is alpha. What is the slope and the confidence interval for our estimate of that slope? So we're trying to make inference. We've simulated knowing it, but we're trying to back out, if these were our data, would we be able to, what would we infer as the slope, as the value of alpha, and with what confidence interval, with what precision? So in fact, if we look at that same fit that we did, so, well, I called it fit before, now it's L model for linear models, the same thing. And we just ask R to tell us about what we found. It's going to, instead of making a plot, it's going to tell us, this is a bit awkward because it's made for terminal, but you'll get used to reading these in the lab a little bit. In fact, this is not showing us okay, so what does this mean? Well, in here, it's showing that it's estimated for that intercept value is 1,000, a little bit more than 1,000, 1,006. And for the slope, which is its estimate of alpha, it's got a -67. Now, this is not showing you explicitly a confidence interval. What it's showing you is a standard error, which is the width of which we can turn into a confidence interval, because a 95% confidence interval is plus or minus 2 standard errors, right? So that means that this is 16. So our confidence interval is plus or minus 30, which means that we would report as a final result that we've estimated the strength of this effect on bees is from something like minus 98 to minus 100 to minus 40, somewhere in there. That would be our conclusion. Okay. And this was a case actually where we had 1,000 observations again. And so that's why we're actually able to resolve this fairly well, right? So it should be clear to you that with just 20 observations, we were not able to resolve this with much confidence. With 1,000, as you can see by this line and the somewhat suggestive predictive confidence intervals here in gray confidence band, we can actually resolve a negative slope, and this is quantifying that estimate. Okay, so in R, we're going to have access to a lot of tools like this one to do different statistical tests and fits and so on. And ultimately, what we're interested in is to estimate some parameter that we're after and to know what its confidence interval is. So the range in which we think that value lies. And that is what statistical power is all about, right? So it's knowing in advance of our design, in advance of carrying out the research, how precise are we going to end up being in our answer? We don't know the answer yet, but we can say something about how precise, how well we're going to be able to resolve the answer. So if we're looking for alpha, somebody may ask in advance, how small an effect are you going to be able to detect, or how precisely are you going to be able to tell me what the size of the effect is? And that comes back to this question that we looked at before, where we have two distributions. In the case of two groups, how small a difference between the means of these two groups are we going to be able to detect confidently? Now, the nice way to ask this research question is, what's the difference in mean bee population between the two groups? We're going to spend some attention on a frequentist statistical approach in which we change questions like that into yes/no questions. And so the hypothesis testing frequentist question corresponding to that research question is there a difference between the two between the means of the two bee populations, yes or no? And then we're going to give some probability of being able to say that there is no difference. Okay, so now I want you to give a general answer given all of the parameters that we've played with in this model today. What does our ability to discriminate between the means of these two groups depend on? Think in terms of the parameters of our design and the parameters in the properties of nature, which we've also parameterized. Okay, so here's a plot of, back to our two groups project design. What is it that affects, and how does it affect our ability to discriminate between the means of the two groups? Take a minute and write down an answer. Complete answer, right? All of the things that affect this. Okay, so coming back, just quickly, we've kind of already gone over this, but number one is the size of the effect that if insecticides, if the chemicals are really bad for bees, then these two distributions are going to be further apart from each other, and it will be easier to resolve the difference between them. The difference will be larger. It'll be easier to resolve because it's larger. Secondly, the scope of that natural variation, which is the width of these two curves, the larger that is, the harder it is to tell a difference between the means. And lastly, the sample size that we have, which is something we can control, the more samples we have, the better we can resolve differences between the two means, right? The more samples we have, the more these curves look like perfect bell curves, and we can just fit a bell curve to each one and say exactly where's the difference between the tops of the two of them. So so more samples will solve our problem here, and with enough samples we could resolve even a very small difference. And again, note, we could resolve a difference that's much, much smaller than the width of these curves. So the width is the variance, in this case, the variance of beehives populations after a year. And we may want to, even if that's 100, the standard deviation of that was 100 in our model, we could resolve a difference between the two groups of only 3 bees if we had enough of a sample size. And I've actually told you the scaling law there is 1 over the square root of the sample size is, in fact, the standard deviation of the natural variation, so that 100 divided by the square root of your sample size, that's the resolution with which you can resolve differences. Okay, I want to now shift gears a little bit to something more general than. In fact, let's just take a quick break here, and come back with part 2, which is going to be talking about something kind of amazing to do with sample size. Okay, so let's step back just a little bit from the scenario, the simulation to do with beehives, and talk slightly more generally about something very amazing to do with sampling. And for those of you who've seen the central limit theorem before, you're going to see something that you might remember. But let me just do a little thought experiment. Okay, so now we have a much simpler scenario. We're simply going to sample, make a measurement of some natural distribution. So pretend that we're sampling the size, we're measuring the size of oak leaves. And here is the natural distribution in the wild of the size of oak leaves. So it looks like the average size is 16, maybe that's centimeters. And the standard deviation here, again, remember, 2 standard deviations is 95% of the population, and so 1 standard deviation here turns out it's 5 centimeters. Okay, so here's all right. Now, what happens if my research design is to go and sample a few leaves and then calculate the mean of my 5 samples? Okay, so here I'm taking 5 samples and I calculate a mean. Let me do that again. I've calculated another mean. Okay, now I want you to take a break and tell me, what is the distribution? Okay, I'm actually going to do this 5 times now. Okay, so I just took 5 more means. I want you to tell me, what is going to be the distribution of these means that I calculate from doing this experiment? So each time I press this animator, each time I take 5 samples, that's 1 research project. I go out, I sample 5 leaves, I calculate a mean, and that's my answer. Down here is the distribution of what happens if people repeat the whole research experiment multiple times. Okay, so what is going to be the shape of this if I keep animating, I keep doing more and more iterations of this experiment? Take a break. Okay, let's keep doing this. I'm doing 5 repetitions of the entire research project here each time. You know what? I'm going to rev this up here, and I'm going to do 10,000 experiments. Okay, here is the distribution. why is that not the same as this? Okay, it is, in fact, look, I've got another bell curve. The oak leaves were shaped, the distribution, the size of the oak leaves was shaped as a bell curve. I've got another bell curve of my inference in multiple iterations of a research project where I'm sampling and taking an average. Now, why is this? Why is it narrower? Note that if my whole research project was just to sample one leaf and record the mean, then I would reproduce exactly this distribution, right? This is the distribution of single leaf widths. But what I'm doing is averaging over a few of them. And so I'm actually zoning in, I'm focusing in a little bit on the correct value. In fact, if my research project, instead of sampling 5 leaves and calculating a mean, if instead I sampled 10 leaves and calculated a mean, so now I'm taking 10 samples, and then I just calculate a mean of those and I record that, and that's my result. Okay, now I actually repeat the whole research project several times. Now what happens, or many times, I end up with an even narrower distribution. And if I'm sampling 25 in each iteration, I'm only going to do that one once before I jump here to a much larger number of samples. Okay, I take 25, I take the average. Wow, the average seems to be right in the middle on that one. And indeed, with 25, my distribution of answers, inference that I end up with at the end of my research project is even narrower. Okay, now, this is actually really deep. Okay, this represents the whole the procedure in all statistical tests which you will do in your stat software, in which we don't just estimate the mean. The stat software is also going to tell you about your confidence interval around the mean. It's going to tell you how confident are we about the value. And the reason it can do that is because it's essentially simulating this procedure, this process. And it says, well, I know something about the width of the distribution if we were to do this many times, right? So if I know something about the width of the natural distribution and I understand my sampling procedure, my data generating process here, I can actually tell you about how narrow, even with that, I don't know, how narrow the possible range of conclusions that I will make, inference I will make, will be if I were to do this experiment many times, and that is the confidence with which I can tell you my answer. Okay, so so that's a major lesson. Now, if that was right, so I want you to remember that the confidence, the width of an inference like this, a mean, is narrower than the width of the variance of what you're observing. But let me show you something weird. What if the distribution of oak leaf widths was not normal, but instead something skewed like this? Okay, so now I want you to whoops, I did not mean to do that yet. I would like you to tell me what the distribution of means is going to look like. The same thing. I'm going to take here now, 25 samples from this. I'm going to calculate a mean. What does the distribution of means look like for this, which is not a bell curve? Take a break, come back, and now let me speed this up. Okay, what do I get? I get again a bell curve, and you can't tell, maybe this is resolution is coarse in this histogram, but in fact, this is again a perfect bell curve. Okay, so this is a super remarkable feature of means, of taking averages of things, that even if this is not a bell curve, the distribution of inference that you're going to make is for something like a mean. Let me just go really weird now. Watch this. Okay, what if the distribution of oak leaf sizes was like this? This is the distribution of oak leaves widths in Montreal, because maybe there are several different subspecies or something. Okay, and what if I do the same thing? I still get, for the mean, I get a bell curve as I do this experiment many times. And in fact, the width of this thing is again the standard deviation of this mess, right? This is not a bell curve, but it still has a standard deviation, 7.7 here, divided by the square root of the number of samples in each of my samples here, which is 25. So, right, so the standard deviation in this thing is going to be 7.6 divided by the square root of 25, which is 5. And so you get, that's about 1.54. By the way, this is true of the mean. It's not true of other stuff, right? If I take the median, the distribution of the median is nothing special. It's very complicated to mapping from the distribution of oak leaves to the distribution of medians of a sample of oak leaves. We can't say anything simple about this. Whereas if we're modeling the mean, estimating means, we have some very nice properties here. Okay, this is the central limit theorem. Okay, and with that, I have one more thing to tell you about. This is some really important language that you need to know, and it's related to precisely what we've been talking about, which is the confidence with which we can resolve estimates, say, of parameters, and of what I mentioned was statistical power at the beginning, right? So statistical power is the power of our method to detect something. So we talk in this language about type 1 and type 2 errors. And one reason I want you to know about this is actually something you may hear about if you listen, you hear news of epidemiological studies on the radio or whatever. You actually may hear about type 1 and type 2 errors. A type 1 error means that I detected an effect. I claimed, yes, I found an effect when in fact there wasn't one. Okay, so we're going to talk about this more as we give some examples of frequentist statistical testing. But this is in the context of these yes-no answers. And so you've heard lots of studies like this. Someone does a randomized controlled trial giving a new medicine or a new diet or something to group A, and you want to know, is their outcome better than the control group B who didn't get the thing? Okay, and so the big question is just detect, is there a difference between these groups? Was the outcome different or better for the treatment group? And a type 1 error means that I conclude statistically that yes, there was an improvement when in fact there wasn't. Meaning that the reason I concluded there was just luck, right? So there's some natural variation in outcomes, and because my sample size was small enough and the size of the natural variation in outcomes was large enough compared with whatever causal effect my new drug or my diet treatment or whatever it is has, my treatment group did better, but in fact not for the right reason. That's a type 1 error. So I conclude that there was an effect when there wasn't. Type 2 error is exactly the opposite, or sort of the opposite. It's when I do a study and I say, look, we did this study and we could not detect a difference. We could not detect an effect. So we gave this new drug to one group and we detected no significant difference between the two groups. And so we have to conclude that there's maybe no effect of the drug. When in fact, there is an effect of the drug and you just didn't have enough statistical power, you didn't have enough resolution, sensitivity, you didn't have enough sample size, etc., to detect it. That's a type 2 error. And so I just have a mnemonic here, if you like. The boy who cried wolf. You hopefully know the fairy tale. This is a child labor story. I suppose the boy is looking after a flock of sheep, and thinks that he sees a wolf, and so he screams for help, help, to all the villagers, come and help me, save me and save the sheep from the wolf. And the villagers come and they say, where's the wolf? And the boy says, oh, I thought I saw a wolf. I don't see the wolf anymore. Boy does this again, no wolf. But boy does this again, no wolf. Boy does this a third time, and the third time there actually is a wolf. And all the villagers say, nah, we're not going to come. There's never a wolf. Okay, that's type 1 error. So it claims to detect a wolf, there's no wolf. prisoner on trial, I think my story here is, that somebody is standing there, the prisoner is in the court, and did in fact commit the crime, but there's not enough evidence for it. And so the jury has to let them go. Jury says innocent because we don't have sufficient evidence to convict the prisoner. Type 2 error, if the prisoner is in fact guilty. Okay, we'll come back a little bit more to these concepts, and next class is going to be looking at some of these frequentist yes-no style tests and more examples. And after that, we do the lab in which you're going to master Control R to be your slave and do all of the work for you.